Red blood cells, also referred to as red cells, red blood corpuscles, hematids. Erythroid cells or erythrocytes are the most common type of blood cell and the vertebrate's principal means of delivering oxygen to the body tissues via blood flow through the circulatory system. RBCs take up oxygen in the lungs, or in fish the gills, and release it into tissues while squeezing through the body's capillaries. The cytoplasm of a red blood cell is rich in hemoglobin, an iron-containing biomolecule that can bind oxygen and is responsible for the red color of the cells and the blood. Each human red blood cell contains approximately 270 million hemoglobin molecules. The cell membrane is composed of proteins and lipids, and this structure provides properties essential for physiological cell functions such as deformability and stability of the blood cell while traversing the circulatory system. In humans, mature red blood cells are flexible biconcave discs. They lack a cell nucleus and organelles. To accommodate maximum space for hemoglobin, they can be viewed as sacs of hemoglobin, with a plasma membrane as the sac. Approximately 2.4 million new erythrocytes are produced per second in human adults. The cells develop in the bone marrow and circulate for about 120 days in the body before their components are recycled by macrophages. Each circulation takes about 60 seconds. Approximately 84% of the cells in the human body are 20-30 trillion red blood cells. Nearly half of the blood's volume is red blood cells. Packed red blood cells are red blood cells that have been donated, processed, and stored in a blood bank for blood transfusion. Description: the erythrocyte is a biconcave disc between 5 and 7.5 micrometers in diameter, 7.1 micrometer thick and 80 to 100 femtoliters in volume. The cell has lost its residual RNA and its mitochondria, as well as some important enzymes, therefore, it is unable to synthesize new proteins or lipids. Their cytoplasm contains mostly the pigment hemoglobin, which gives them their characteristic red color and is responsible for oxygen transport. However, this description applies to mammalian erythrocytes, since in the rest of vertebrates, with some exceptions, erythrocytes lack the biconcave shape and are usually larger than those described above. This is because the red blood cells of the rest of the vertebrates still have a nucleus. Red blood cells are derived from committed stem cells called hemocyst cells. Erythropoietin, a growth hormone produced in renal tissues, stimulates erythropoiesis and is responsible for maintaining a steady-state red cell mass. Erythrocytes, like leukocytes, originate from the bone marrow. The erythrocyte concentration varies according to sex, age, and geographic location. Higher erythrocyte concentrations are found at high altitude, in males, and in newborns. Decreases below the reference range lead to a disease state called anemia. This alteration causes tissue hypoxia. Increased red blood cell concentration is less common. Hemolysis is the destruction of aged red blood cells and occurs in macrophages of the spleen and liver. The essential elements, globin and iron, are preserved and reused. The heme fraction of the molecule is catabolized to bilirubin and biliverdin and is ultimately excreted through the intestinal tract. Intravascular rupture of the erythrocyte releases hemoglobin directly into the blood, where the molecule dissociates into alpha and beta dimers, which bind to the transport protein, haptoglobin. This transports the dimers to the liver, where they are subsequently catabolized to bilirubin and excreted. In addition to their role in oxygen transport, erythrocytes are also important for maintaining an adequate concentration of iron in the body. Iron is needed to produce hemoglobin, and a lack of iron can lead to a condition known as anemia, which is characterized by a decrease in the number of erythrocytes and hemoglobin in the blood.